Hello, I'm Philippe Grandjean. I'm a physician and professor of environmental medicine at the University of Southern Denmark. I'm also an adjunct professor at Harvard University. I'm very pleased to be with you today because you are in Grenoble, the green city of this year, and you are discussing the call of Grenoble that is going to tackle the difficult problem of syndemics. And so I know that the réseau environnement santé are also involved here. So what I want to share with you is my experience with syndemics that are associated with the perfluorinated compounds. And I'll show you my slide here that illustrates what the perfluorinated compounds are. You can see it's a carbon chain that's completely enveloped with fluorine. And these compounds have this property that was thought to be very useful, namely they are persistent, they don't break down. But unfortunately, they accumulate in the environment and they accumulate in the human body. You have them in your body. I have them. And I mean, basically, I have never met a blood sample, a human blood sample, that did not contain these compounds. They are over the whole planet. They are everywhere. So what I wanted to show you is a brief slide that illustrates how they disperse in the environment. They end up in sewage water. They get into the sewage sludge. The sludge is spread on agricultural fields. The water runs out into the ocean and where it pollutes the marine food. So one way or another, these compounds end up on our dinner plate just about every day. We are constantly exposed to them. And you might ask the question, but how come this is big news? We've been using these compounds for many decades. Well, the point is that it was actually discovered way back in time that these compounds result in exposures to the general population, and of course also to workers, and they disseminate. They were even found in the albatrosses. And so this knowledge emerged very slowly, and in particular, part of that knowledge was kept secret by the producers, and we didn't know until the beginning of this millennium that this information had actually been obtained. The same regards the immunotoxicity of these compounds. It was demonstrated first time in 1978, and we didn't know. We were not told. So our recent insight into immunotoxicity was simply a result of ignorance, the result of secrecy. And that there are many ways that one can perhaps counter those problems, but certainly it has materially affected our research insight. It has delayed us. But I wanted to just briefly show you a simplified model of the immune system. There are various cells involved. And when we get infected, then that pathogen will act as an antigen on some of these cells, and they will spur the development of plasma cells that will develop antibodies. Now, that is exactly what we want to obtain by vaccinations. And the World Health Organization has supported the idea that to document immunotoxicity, we should use antibody responses to routine vaccinations for reasons that are outlined here. And this is exactly what we have done over the years. But our first study actually targeted adults, my 12 colleagues, medical colleagues from the National Hospital in Copenhagen. And as you can see here, we took several blood samples from them and measured the antibody concentrations after their tetanus vaccination. And you can see that the responses differed a little bit. And actually, one of them had virtually no response to the vaccination. So we wondered why were these curves so different? Some of them were steeper than others. And we found out that the steepness was actually associated with the serum concentration of the PFAS. So the more PFAS my colleagues had in the body, the flatter the curve. So we wanted to set up studies in children, and this is just a model of when the vaccinations, the routine vaccinations occur, and they result in antibody increases. And we took some blood samples, and the major finding was that with increasing PFAS exposures, then the antibody concentrations were much lower. In fact, the children would lose about 50% of the antibody concentration for each doubling in the PFAS concentration in the blood. So where does this PFAS come from in small children? Well, there's unfortunately a very simple response to that. It is transferred through the placenta. So the fetus shares the mother's blood concentration of these compounds, but they're also eliminated into human milk. So here you can see that the longer a child has been breastfed, the more buildup of these compounds happens in the child. And a child that has been long-term breastfed may develop something like a 10-fold increase in the serum concentration above what the mother had. And so we are that way exposing the next generation at the very, very vulnerable age just after they're born and the organ functions are developing rapidly. And as you can see on this slide, EFSA actually mapped the lifetime occurrence of PFAS in the blood, and it's very clear that that there is a peak here at the completion of breastfeeding. A very ironic and sad evidence that maybe human milk is no longer the most nutritious way of supporting a baby with nutrients and vitamins, etc. It's being contaminated by some of the worst pollutants that mankind has developed. Now, this just illustrates that the peak occurs when the adaptive part of the immune system develops, that is, during infancy. So how does that relate to COVID-19? Well, we know that those infections are particularly serious in the elderly and in men, and also in subjects who have pre-existing diabetes type 2 or obesity. Now, in regard to the PFASs, we know that the elderly and men have higher accumulated burdens of the PFASs in the body. But we also know that elevated exposures to PFASs are associated with an increased risk of developing diabetes and obesity. So it's like there is an association here in the causation. So the study we designed consisted of a little over 300 subjects who had tested positive for the coronavirus and who had blood samples taken for any reason in connection with, in a time connection with a corona positive test. And then we got anonymously, we got information on the course of the disease so that we could detect whether PFAS exposures were associated with the severity of the COVID-19 infection. And in fact, one of the PFAS is the one called PFBA, the one that has four carbons, the butane congener, and you can see that there's a very strong association between the concentration of this compound in the blood and the severity of the COVID-19 disease. But why was it this particular one? Well, perhaps there's an answer here from a Spanish study of that documented that that is exactly the PFAS that concentrates in the lungs. So it's very likely that the toxicity expressed by the PFAS was actually particularly associated with that PFAS because it concentrates in the lungs, where the SARS virus is also causing its most damaging effects. So overall, I would say we know that exposure to these compounds result in a lower antibody response. We also know from other studies, we know that children with elevated exposures more frequently have high fever. They get more infections than other children. We also know they get more hospitalizations for infectious disease, particularly lower airway infections, pneumonia. And there are also associations with the COVID-19 that we're working to support or refute, so that's still being investigated. And we certainly know that there are other outcomes possible. The World Health Organization has suggested that the association with cancer from PFAS exposures may be due to the induced deficiencies of the immune system, because the immune system is also hopefully destroying cells that become abnormal, and also cells that could potentially generate cancer disease. And when we present evidence like this, then we have to realize that there are some biases toward the null. We are often underestimating what's really going on. And for reasons like this, one of them being that we always have imprecise exposure data. We may just have one blood sample to measure the exposure, and that does not characterize the full lifespan exposure of that subject. But anyway, there are various weaknesses of the studies that we do, and oftentimes I hear the argument that if there is no statistical significance, it means it's a negative study. It argues against, supposedly, that there is a causal relationship. And that is, of course, completely nonsense. But that is an argument that I'm sure that you are aware of and that you have heard. But because of those kind of concerns, then we have been very slow in generating proper protection of the population against these compounds. For example, when they pollute the drinking water. And as you can see here, the first exposure limits were very high. And as late as 2020, the EU decided for a total limit of 100 ppt, that is 100 nanograms per liter of drinking water. But if you take the risk assessment done by the European Food Safety Authority from the same year, then the maximum intake limit that EFSA agreed on corresponds to a much lower drinking water limit of 2 nanograms per liter. So you can see we are behind. We're behind in translating science into policy, and we are behind because science has been delayed and it has been underestimated. So I have some conclusions that illustrate that the need for you to develop the call of Grenoble to highlight the significance of syndemics. And as you can see here, we have toxicity that affects all of us. The background exposures are toxic. They are interfering with our immune system responses. So we also need to make sure that we protect children in the fetus, because they're much more vulnerable than we as adults are. So because of these syndemic consequences, I think that the perfluorinated compounds, the PFASs, are a very good example of the need for you to develop the most convincing statement on behalf of the city of Grenoble. I'm sorry I couldn't be part of that, but I wish you the best of luck and thank you for allowing me to speak.